Okay, so I guess most people are familiar with psychedelics as acting in the brain and as now really exciting therapies for mental disorders. And in my laboratory, in my research, I have been studying a lot of the neuroscience of psychedelics. But we discovered in my lab several years ago that psychedelics are really potent anti-inflammatories. So about half of my research is now devoted towards developing and understanding how psychedelics are anti-inflammatory. So I'm going to talk to you about that today. My disclosures, our lawyers make me present. So you probably all are familiar with psychedelics. They come in many different varieties from nature. The classic psychedelics like mescaline from the peyote cactus, psilocybin from many species of mushroom, DMT, which is the active ingredient in ayahuasca, 5-methoxy-DMT from toads, and the precursor for LSD, lysergic acid, comes from this fungus that grows on rye. The chemicals for each of these fall into 3 general chemotypes, and they're all very structurally related to serotonin, shown here on the bottom left, 5-hydroxytryptamine. Tryptamines like DMT, psilocin are very similar in structure to serotonin, and ergolines have the structure of serotonin embedded within, so it's like with LSD. And the phenethylamines, which are related to mescaline, shown on the right, also have some similarity to serotonin. So the behavioral effects of psychedelics are primarily due to activation of the serotonin 2A receptor. There are, I think, 14 different receptors for serotonin in mammals. The serotonin 2A receptor is what's called a G protein coupled receptor. And there's high expression in the brain, especially in frontal cortical areas, shown here on the right. And engagement of this receptor has been shown both in animals and in humans to be necessary and sufficient to produce behavioral effects from psychedelics. But the serotonin 2A receptor is actually, it's the most widely expressed serotonin receptor throughout the body. It's been found in nearly every cell type and tissue, heart, vascular system, the GI tract, immune system, bones, muscle, eye, skin, and it is primarily mediating an inflammatory response from serotonin. But we found that certain psychedelics, not all, are actually potent anti-inflammatories at the serotonin 2A receptor. And we have been primarily looking at peripheral inflammatory related disorders and diseases and tissues, although now we're starting to look in the brain at neuroinflammation. So how is this possible, that serotonin is pro-inflammatory and psychedelics are anti-inflammatory? Well, the G protein coupled receptor sits in the membrane and it bounces around like this. It's in a very dynamic state. And serotonin engages with a particular subset of amino acids in this receptor to stabilize it, to recruit effector pathways, primarily G-alpha-Q, that perform these essential cellular functions. And there's some evidence that psychedelics recruit different effector pathways or through interactions with slightly different subsets of amino acid. And anti-inflammatory effects, we believe, are the drug binding to in a very specific way to certain amino acids in the binding pocket that couples the receptor to anti-inflammatory pathways that serotonin does not. So this is a close-up of a molecular dynamic simulation that we have done in my lab, showing a molecule of 2CB in the orthosteric binding pocket. So you can see that these interactions over time are very dynamic. They're shifting back and forth between different amino acids, and there are some amino acids that are being engaged more than others. And depending upon the structure of the psychedelic, it will engage these different amino acids to different extents to activate the receptor. So in some of these activated states, it induces a potent anti-inflammatory effect. So one of the main lines of research in my laboratory is trying to understand how these interactions will, on one hand, lead to behaviors, and then on the other hand, influence inflammation. We first discovered the anti-inflammatory effects using a drug called DOI, which is 2,5-dimethoxy-4-iodoamphetamine, which is shown here on the left. And it's a phenethylamine psychedelic that's related in structure to mescaline, and it's very highly selective for activating 5-HT2 receptors. Tryptamines like psilocin and DMT pretty much activate all serotonin receptors, and ergolines like LSD will activate about all serotonin receptors, plus many additional monoaminergic receptors like dopamine receptors. But DOI is very selective for 5-HT2 receptors. It's not used really recreationally because it produces about a 30-hour subjective experience. And it's proven over the last 30 years to be about the most important pharmacological tool to really study 5-HT2A receptors. One of the early experiments we did was to just pre-treat mice, normal wild-type mice with DOI, and then induce systemic inflammation with a moderate amount of TNF-alpha, which is an inflammatory cytokine-type protein. And it was enough to induce a systemic inflammatory effect that would normally subside after a few hours. It wasn't a very large dose, but it was enough that we could look at inflammation. And after 5 hours, we looked at different tissues to see what the anti-inflammatory effects were. And some of the results are shown here from the small intestine on the left and serum IL-6 on the right. And these are two a cytokine IL-6 and MCP1, which are induced in tissues where there's inflammation. So the higher the bar, the more inflammation. In the TNF treated animals with tumor necrosis factor alpha, you can see that there's this very large increase in the expression of these genes. And in the presence of TNF alpha and DOI, even at very low levels, we substantially block this inflammation in the intestine. And in the serum, we looked at the protein for IL-6, and you can see again a nice dose response here for blockade of this inflammation induced by TNF. And the largest dose of TNF that we used here, 0.3 milligrams per kilogram, is about the behavioral threshold dose of DOI in a mouse. So at these lower doses here, 0.01 milligrams per kilogram, this is far below the minimal behavioral threshold. Another model that we looked at was in cardiovascular and metabolic disease, which is a knockout mouse model for the ApoE gene. This mouse, when it's fed what we call a high-fat Western diet, which is essentially high cholesterol, high fat, it will develop severe atherosclerosis and cardiovascular disease over the course of a few months. And what we did with this model was we used these osmotic mini pumps that slowly release drug continuously over time, so we don't have to keep injecting the animal every day. And we can essentially program this to release a specific amount over time. And it was a sub-behavioral, I would say, if you're thinking microdosing, we are essentially microdosing these animals with DOI. And then we fed these mice a high-fat diet, and then we looked at the cardiovascular disease. And two of the main outcomes that we looked at were, again, this inflammatory cytokine IL-6 and another cytokine called vascular cell adhesion molecule 1, or VCAM1. And you can see the higher bars are the increased expression. So these demonstrate the inflammation. In the groups where we just fed them normal food, not the high fat, high cholesterol food, there was not really any increase in inflammation, and the DOI didn't have an effect to suppress any of the baseline expression. But what you can see is that these animals that were fed high fat, the DOI completely suppressed the expression of these two cytokines. And we saw this with others as well. So we're having a very potent anti-inflammatory effect on the vascular tissues. So this suggests that it could potentially be used as a protective for cardiovascular disease. Another thing about this model is that it's also a model for diabetes. And what we showed is that in these same animals, high fat fed DOI, that they were able to utilize blood glucose more efficiently. So it protected against the diabetic response as well. The primary assay and model that we've used over the last, I would say, 10 years has been allergic asthma. And we study asthma not because really we want to cure asthma or understand asthma, but because asthma is an inflammatory disorder that involves almost all aspects of the immune system. It involves non-immune tissues like smooth muscle cells, endothelial cells. It involves immune cells like macrophages and T cells, T regulatory cells, which their discovery was won the Nobel Prize last week. So this has been a very powerful model, and we use a variant of this called the ovalbumin model. And this is a model of allergic asthma. So think of the type of asthma, say it's there are flowers or trees or grass that are blooming outside. Somebody will go outside, they'll expose themselves to the pollen, and suddenly the airways will develop inflammation and constriction. And we generate animals with allergic asthma by sensitizing them with ovalbumin, which is the chicken egg white protein. And we put them in a chamber for 20 minutes to breathe this nebulized ovalbumin, and it induces a allergic reaction in the lungs that is very conserved with the physiology of asthma. And when we want to test a drug in this model, we use a nebulizer like this, and we have a little nose cone, and we take the asthmatic mice or rats, and we put the nose cone on them and give them essentially nebulized inhaled drug, like a human would do with an inhaler to treat their asthma. And with this model, we can measure pulmonary mechanics in alive, awake animals. And that's been the primary outcome that we look at as a proxy for the pulmonary inflammation. And this is one of our very first data sets that was published 10 years ago. And this is from a mouse, and it's an airway in the lung of these mice. And you can see here on the very left, the far left, an airway of a normal lung. This was an animal that was not treated with ovalbumin. And below it is sort of a wider view of that lung tissue. The set to its right of images are what the asthmatic mice lungs look like. The airways are very thick, and they have produced an excess of mucus, which is staining pink in the top here. And in the bottom is a different kind of stain that highlights immune-related cells, like macrophages, T cells, and it forms this peribronchial infiltrate, which is the pulmonary inflammation of the lungs. And you can see that there's a lot of inflammation around these airways. And on the right is an animal that was pre-treated with a very small amount of DOI by an inhaler. And you can see that it completely prevented the development of both mucus and pulmonary inflammation, and it restored normal breathing to these animals. And we found that at even a dose 10 times less than this is very effective at preventing the pulmonary inflammation. So we found that we could prevent pulmonary inflammation. What about rescuing it? So we developed a model of severe asthma by continuously treating with ovalbumin over several weeks um, to have these animals with established asthma that we verified over a couple weeks, and then we treated with DOI. These are lungs from that chronic model. And what I would like to point out here is in this chronic model, this green staining represents collagen. So this is fibrosis that's happening in the lung that in individuals with severe asthma, not only do they have inflammation, difficulty breathing, but the lungs become fibrotic and the airways become stiffer. And this contributes to the pathology of the disease. And in these animals that have thickened airways, fibrosis in the lungs, we then treated with, it was 3 consecutive days of inhaled DOI for a particular dose. And what we saw was pretty remarkable. We had a 70% reversal of the fibrosis. The pulmonary inflammation went away, and the breathing of these animals, when we tested them, was just like the control animals. So we can not only prevent inflammation, but we can also rescue inflammation with DOI. So at this point, we were wondering, was this something that was specific to the drug, or was it applicable to all psychedelics as a class? And we looked at the anti-inflammatory activity of about 25 different drugs that were all primarily psychedelics. And that is shown here, rank ordered from those that had no anti-inflammatory effects in the orange-pink color to those that were fully efficacious as an anti-inflammatory. And this dotted line in the middle represents sort of the average marker of a asthmatic mouse. So it's a measure of PEN-H. It's an arbitrary unit, but it's an aggregate measure for pulmonary inflammation. So the higher the bar, the more pulmonary inflammation and the more difficult time these animals are having breathing. And this was done in rats. And we found that a drug like DMT and 5-methoxy-DMT, which are clearly very powerful psychedelics, did not really have any anti-inflammatory effects. LSD, which is arguably one of the most potent psychedelics known, even at relatively high doses, was not able to fully rescue the inflammation in these asthmatic animals. But other drugs like DOI over here and psilocin were fully able to have an anti-inflammatory effect in the lung. So we looked at signaling pathways that underlie the behavioral effects that are primarily GQ and beta-arrestin, although there's some debate on what role both of these play. We looked at both of those and saw that there was no correlation between the signaling at GQ or beta-arrestin or behavioral activity in a head twitch response. And so this data altogether told us that the signaling pathways and effectors that these certain psychedelics recruit to be anti-inflammatory, we can likely separate through medicinal chemistry away from the behavioral effects. So one of the things that we've done in my lab, not going to really show any of that data here, but we've taken some of these I call them first generation, like DOI, and we've, through understanding how it interacts with the receptor, have made modifications to where we can maximize the receptor activity and anti-inflammatory effects, but we have no head twitches, which is a behavioral measure for psychedelics. So as part of that, these are some of the different drugs that we've used, and we looked to see what types of modifications were allowable. Those are shown in green with the phenethylamines. What kind of modifications that inhibited the or reduced the anti-inflammatory activity. Those are molecule derivatives shown in yellow. In the ergolines, we have not really found an ergoline that is a full anti-inflammatory agent. And the tryptamines are very interesting, that when we look at psilocin, and you can see down here the structure of this tryptamine, it's dimethyl N and dimethyl 4-hydroxytryptamine. If we remove this hydroxy group here and convert that to DMT, we've gone from a full potent anti-inflammatory to no activity at all in our assays. And we verified this with a similar drug called 4-hydroxy-DIPT. And when we compare that to DIPT, there's no anti-inflammatory efficacy. So this kind of knowledge allowed us to go in and do some molecular dynamics and modeling in silico to develop some next generation anti-inflammatories. One of the more interesting compounds that came out of this analysis was this drug DOTFM. And it's very closely related in structure to DOI. And the only difference is DOI has this iodine at the 4 position, where DOTFM has this trifluoromethyl. These have virtually identical pharmacology at the receptor. The affinity for the agonists, they both induce head twitches very potently, but DOTFM is not anti-inflammatory in any of the assays that we've looked at, both in vivo and in vitro, whereas DOI is. So what we attempted to do was use DOTFM and DOI as tools to try to identify what the mechanism is. What is DOI doing differently than DOTFM? And not only is DOTFM not anti-inflammatory, but it's pro-inflammatory in some of our models. So it's acting more like serotonin, whereas DOI is acting more like psilocybin or psilocin or some of the other anti-inflammatory compounds. And we did this. One of the first tests we did was looking at lungs and looking at the protein response in quantitative proteomics. So we can take the proteins from the lungs of the different treated animals, and we can analyze them to see which proteins were differentially expressed or induced or repressed in expression by which drug. And our original hypothesis was that if we looked at the subset of the set of proteins that were altered in expression by DOI and compared to DOTFM, that there would be a large overlap because they're both very good identical agonists at the 5-HT2A receptor with respect to just regular in vitro pharmacology. But this was not what we saw. There was a very dramatic difference in the proteomic response between these two drugs. In proteins that were increased by these drugs, there was only a 24 protein overlap. And in drugs that were decreased, again, there was only a fraction of those were overlap between the two drugs. So we published on this these results, I think, last year. And one of the more interesting proteins that we found that DOI changes expression of that DOTFM does not is this protein arginase 1. Here you can see this is what's called a volcano plot, where we're looking at the blue dots are proteins affected by DOI, either increased or decreased. And the orange dots are proteins that are affected by DOTFM, either increased or decreased. And the ones that are significant are shown in these not grayed out quadrants. If they're grayed out, this is not really considered significant. So arginase 1 is an interesting protein that has been linked to asthma and many different inflammatory processes. It's an enzyme that metabolizes the amino acid arginine. And when it's present, arginine is shunted through this arginase pathway to form these other intermediates, which ultimately can produce fibrosis and inflammation. And in the absence of arginase, arginine is metabolized through a different pathway involving nitric oxide synthase to produce nitric oxide, which can produce an anti-inflammatory effect. And we've seen this in some of our other models as well. Many of the other proteins that DOI has change in expression, some of those are involved in epigenetic factors, especially in modulating histamine and chromatin structure. When we did an analysis of the proteins, the DOI had a highest significance for modulating proteins that were involved in metabolic pathways. So these are subsets of proteins that are significantly increased and decreased. So metabolic pathways were the most changed. And for DOTFM, we also saw metabolic pathways that were most changed. But the nature of these metabolic pathways were very different. And these are interaction networks here on the left of some of the proteins and how they're interacting. These So another way to look at these proteins in metabolic pathways are this kind of analysis, which is a KEGG pathway analysis. So imagine each one of these nodes is a known defined protein, and all of these pathways represent the interaction of these proteins in metabolism. This circle down here in blue is the citric acid cycle, for example. Each one of these dots here represents a protein in that cycle. And the pathways shown in blue are the pathways that are predicted to be affected by the proteins influenced by DOI in the asthmatic animal. And if we do the same type of analysis with the proteins from DOTFM, we can see that there's a very different set of metabolic pathways that are changed. So that we are continuing our studies in asthma, doing single cell nuclei sequencing, comparing DOI and DOTFM to look at individual cell types now. Another model that we've looked at more recently is rheumatoid arthritis, and this is in collaboration with my colleague, Dr. Kim Mix at Loyola University in New Orleans. So rheumatoid arthritis is when there's inflammation of the synovium, and it can produce chronic inflammation and be very painful and disfiguring. And this can be modeled in mice, and in particular a transgenic mouse that overexpresses human TNF alpha. This mouse begins life fairly healthy, and at about 10 to 12 weeks of age, it will start to develop the symptoms of arthritis, and by 20 weeks of age, the joints are fairly arthritic. And so what we did with this model was we tested 3 different drugs, DOI, very closely related to CB. It's just missing this alpha methyl here. This bromine and iodine are fairly equivalent pharmacologically. And then psilocybin, which is the prodrug of psilocin, which this would be, this phosphate is cleaved off to a 4-hydroxy. And we essentially microdosed these mice with a subbehavioral dose every day from Monday through Friday, gave them 2 days off over the weekend. And every week we tested various measures of joint thickness, paw thickness, the arthritis score. And over the endpoint of the experiment, the area under the curve is shown here for the total arthritis score. Control is the arthritic animal, and we have the 2CB, the DOI, and the psilocybin given at subbehavioral doses every day, Monday through Friday, for weeks 12 to 20, had an anti-inflammatory, anti-arthritic score. When we looked at paw thickness, this is measuring the paw thickness, the swelling that occurs. We use this particular gauge here to measure the thickness of the paw. This control blue is the saline control treated arthritic animal. We see a significant reduction in paw swelling with 2CB, but we did not with DOI or psilocybin. And this orange bar over here on the right is a wild type littermate. So this is a littermate that does not have that human TNF alpha gene. So it is considered a normal wild type mouse. And 2CB almost reduced the thickness back down to these non-arthritic levels. We saw effects on different markers of inflammation when we looked at the expression of genes in the paws of these animals. And one of these we looked at was NF-kappa B, which is a master regulator of the TNF alpha signaling inflammatory cascade pathway. And another one, ICAM1, intracellular cell adhesion molecule 1, which is highly linked to inflammation. And the wild type, you can see it in the white bar here. This is the saline control in the blue bar, which are the arthritic animals not treated with drug. 2CB reduced the expression of both of these significantly. DOI reduced it, as well as psilocybin, but not to the same extent. We see all three reduced ICAM expression over here. And we've looked at, I think, about 8 or 9 different proteins so far. And the general trend among these proteins is that 2CB seems to be very good at blocking all the inflammation that we've looked at in these animals. DOI is not quite as good as 2CB, and psilocybin has been much poorer. It's only able to really produce this significant decrease in about half of the pro-inflammatory markers that we've looked at. So there's some kind of functional selectivity within this model with these different drugs. And all of these drugs were very effective anti-inflammatories in our asthma model. So what we've come to appreciate is that although these psychedelics have potent anti-inflammatory activities, they're probably inducing more than one type of anti-inflammatory mechanism in the cells. And depending upon the nature of the inflammation in that inflammatory disease, some drugs might be functioning better than others. And we looked at the 2CB treated animals and did morphological scoring of the bone. And this was in one of the joint tissues. We have the normal, this is a wild type mouse on the left. The arthritic mouse, you can see kind of this pink structural organization here. That's the bone degeneration. This is where inflammatory cells have come in and degenerated the bone, which is this more uniform pink color. And in the arthritic animals treated with 2CB, you can see that there's about a 50% reduction at the endpoint for the bone degradation and degeneration in these animals. So 2CB has proven in these experiments to be a fairly effective anti-inflammatory and potential therapy for arthritis. Okay. Another feature of this animal is that TNF alpha is also one of the main causative factors in the pathophysiology of inflammatory bowel disorder. So this mouse is also a model for IBD. And we took the intestine from these animals and also probed them for the expression of these pro-inflammatory proteins. This shows a subset of those 4 proteins. Again, here you can see 2CB significantly reduces across the board. DOI reduces in some, but not all. And psilocybin is really only effective in 2 out of 4 of these. So again, as we saw in the pause, there's this trend for 2CB to be much more effective than something like psilocybin in this model. Another thing I'd like to point out is one of the unique features that we found not only in this model, but in the asthma model and the cardiovascular model and some of the cell-based models that we're looking at is that the psychedelics aren't acting like an immunosuppressant. If we have a healthy animal or a cell that's not stressed and we give up to even a relatively large amount of, say, DOI or other anti-inflammatory psychedelic, we don't repress these pro-inflammatory markers below baseline. If we were to give a corticosteroid to these animals, we would see a repression of something like ICAM below the control levels. And we have never really observed this in our models. So we think that these calling them anti-inflammatories is not quite the right terminology, but they are acting to return the systems to homeostasis. That when a system becomes abnormal or inflamed or stressed, produces these markers of inflammation, both in immune cells and non-immune cells. And then somehow the inflammation primes those 5-HT2A receptors to receive the signal from some of these psychedelics as anti-inflammatory instead of pro-inflammatory. And when that signal resolves, that inflammation goes away, then the drugs don't really have any effect. So they kind of only work when you need them to work as an anti-inflammatory. So a summary so far is we've got proof of principle data for anti-inflammatory effects in vascular inflammation like atherosclerosis, metabolic dysfunction like diabetes. We've looked at 2 different models of inflammatory bowel disease now, asthma, rheumatoid arthritis, and my colleague at LSU down the hall has been studying ocular inflammation with these drugs and has found some very potent anti-inflammatory effects with ocular-related inflammation. Some of the interesting features about 5-HT2A receptor agonist therapies that they're very potent. They're efficacious at sub-behavioral levels, oftentimes 30 to 100-fold lower than the threshold behavioral dose is sufficient to produce a significant therapeutic effect. They're steroid sparing, so they're not a general immunosuppressive, so it leaves the immune system intact. And this could mean fewer side effects once translated to the clinic. And it's context dependent. We don't see any anti-inflammatory effects in a non-inflamed or non-diseased state. So one of my passions has been the fruit fly, Drosophila. I actually got my PhD in studying the fruit fly and neurodevelopment in the fruit fly, particularly of the development of the retina in the fruit flies. And I've continued to study fruit flies throughout my career because I think they're a really interesting model that we can do some interesting genetics and other things with. The brain of the fruit fly is fairly complex. There are over 100,000 neurons in these specialized regions that mediate very complicated behaviors like aggression, courtship, flight navigation, learning and memory. And it shares fundamental neurochemistry with mammals. There's serotonin, there's dopamine, there's glutamate, there's GABA, there's histamine. But the main feature is very sophisticated genetics that we can do with fruit flies that we can't really do with mammalian systems. And during my, this image was captured during my evacuation after Hurricane Katrina, and it highlights the 5-HT2A receptors in the fly brain, that they're fairly spread out in all of these different regions. So the target of psychedelics is in the fly. And I've done a lot of work characterizing the effects of psychedelics on behaviors in the fly. But more recently, we wanted to look at inflammation in the fly and neuroinflammation in the fly. And we've found this report from 2013 from Barry Ganetsky's group at Wisconsin, where they had developed a device for looking at a closed head model of traumatic brain injury, which they demonstrated induced inflammation, neurodegeneration, and we have adapted this and got this going in my lab, where essentially we have a spring with a vial with some flies in it, and then the spring is released and bangs into this styrofoam pad, and the flies bounce around this chamber, and they develop essentially neurological symptoms. They have coordination issues, they have neurodegeneration, and they have neuroinflammation. So we have looked at the fly equivalent of pro-inflammatory cytokines. We've looked at two of them. One is called ATC and the other DPTB. The fly has an innate immune response. This would be the equivalent of macrophage immune response in mammals. And the height of these bars represents the increase in expression. After the TBI protocol, you can see both of these have fairly large, robust induction in their expression. And when we feed these guys, pre-treat with DOI, so we keep the flies on food with DOI for 2 days, and then we do the protocol, and then wait 24 hours, and then take the fly brains and do the gene expression analysis. And for both of these pro-inflammatory cytokines, there's a nice dose response with DOI in the food to suppress this neuroinflammation induced by the TBI protocol. And we've replicated this several times with DOI, but with psilocybin, we don't really see an anti-inflammatory effect. And that could be because psilocybin could be acting at other receptors in the fly brain that could potentially be blocking the effect. So with that, I would like to acknowledge the current and former people in my laboratory who have contributed to a lot of this research, the support, financial support, 2A Biosciences, River Styx Foundation, donations, American Asthma Foundation. Over my career in studying psychedelics, the NIH has been not really amenable to funding psychedelic research. Fortunately, that's changing. So most of my funding has been from private foundations and biotech companies. But there have been new grants from the NIH over the last couple of years that have funded some very interesting psychedelic studies. So hopefully that will continue to increase. Okay.
Thank you. Thank you so much for
There we are.
really interesting presentation. I guess I will just, I have some questions as well, but let's jump to questions. Then I'm not sure whether people would like to just talk or rather type in the chat. Just go ahead.
Yeah, I saw one hand raised, Laetitia.
Yeah, but it's gone. Ah, yes, Louis, maybe?
Hello, do you hear me?
Yes.
Yes.
Okay. Obviously, inflammation is a bit of a complex topic, but yeah, there's the simple version in my view is like there's a stressor, and then there's a bodily reaction. I'm curious because you mentioned that the psychedelic anti-inflammatory effect didn't decrease the inflammation markers below like the baseline threshold,
Right.
which steroids would do, for example. So my question then is is there an effect that's lasting, like after the dose is given, or is it somehow a learning effect, or does the treatment need to continue for the effect to continue?
Yeah, that's a very good question, and something that we've been interested in looking at and haven't really fully explored it yet, because these particular models are expensive and labor intensive. So we've just focused on some of the more acute effects, but we do see epigenetic markers. So we might see more lasting effects, such as what's seen in the treatment of depression, but we haven't really looked at those yet.
Okay, thank you. Exciting.
Okay, so I see more people raising their hands. Maybe Andrea?
Thanks. Excellent presentation, excellent research. So that was very nice to see. I might have missed a piece, but I was wondering, is the potential anti-inflammatory activity of psilocybin and other psychedelic linked to the binding of the 5-HT2A receptor, or linked to something else, like the expression of genes of other proteins? What's your view?
I think it's more related to the effector pathways that it's coupling to. That we're changing we're engaging certain residues in that receptor, making a slightly different conformation than serotonin, which is enabling anti-inflammatory pathways to couple. Because we see some drugs that have very weak affinity for the receptor will have full anti-inflammatory efficacy at the higher doses, like a drug like 2CH will. But then a drug like LSD, which is very potent at the receptor, and DOTFM, which is also very potent at the receptor, I think it's sub-nanomolar affinity, we don't see at that. So I think it's really at the signaling level.
Right, thanks.
Quentin?
Yes, hello. Thank you for a fascinating presentation and fascinating results, even though I don't have the scientific background to understand as much as I would love to. I have a pretty naive question. So you said serotonin is pro-inflammatory. Would that lead you to say that substances such as MDMA, which release a lot of serotonin in the brain, would they be considered pro-inflammatory in mice models, for instance?
Oh, that's a complicated question. I think with serotonin being pro-inflammatory, it's at when inflamed tissues are looked at, there's increased serotonin, and when serotonin is added to these tissues, there's more inflammation that occurs at the very simple level. In the brain with something like MDMA, the serotonin transporter itself has, when it's active, can signal and be anti-inflammatory. This is a lot of work that Randy Blakely has done, who's in Florida, is looking at activation of the SERT can decrease IL-1B in the brain. So there's several pathways that are occurring, and we have not looked at anything like MDMA in terms of its pro or anti-inflammatory effects. That's something that we've wondered about, but we haven't looked at that drug yet.
All right. Perfect. Thank you. Yeah.
Then Alexander and Rebecca.
Yes, hi. Thank you for the wonderfully fascinating talk. I was wondering whether there's a similar desensitization or sort of tolerance that occurs to the anti-inflammatory effect, similar to as we experience the behavioral effects less and less with repeated use.
Yeah, I don't think so. In the studies that we've looked at, like for example the cardiovascular disease model, where we had that osmotic mini pump slowly releasing over time, we had a very profound anti-inflammatory effect. in the mouse at arthritis model, we were giving an injection every day. But then, just in case, we gave them 2 days off because we weren't necessarily sure about that. But my theory is that you won't see the tolerance because the dose is so low that the tolerance relies on internalization and desensitization based on receptor occupancy. So if you have, I think Gita Knudsen has shown in her lab, you have to have around a 65% receptor occupancy to produce that peak psychoactive state. And so that's engaging and activating a lot of the receptors, causing that internalization and desensitization. And the amount of receptor that we are activating is probably less than 5 or 10%. So it's not enough really to trigger that process, but we haven't specifically tested that. But my best guess is that we won't see it because the doses are so low.
Okay, that's excellent news. Thank you.
I think Louis might have another question.
Yeah, I have one more. I was wondering about the pharmacological profile of these drugs. Like, do they all have the capacity to cross the blood-brain barrier, or is there differences there too?
Yeah, they all get into the brain fairly well. They induce head twitch behaviors. Like DOTFM and DOI are very, very good at inducing head twitch behaviors. Drugs like DMT, they produce fairly robust head twitch behaviors. So it's not, I don't think it's really a blood-brain barrier penetrant issue. Part of the sort of in our medicinal chemistry and drug design in trying to come up with, say, maybe a peripherally restricted anti-inflammatory, it might get more favorable review at the FDA, even though our doses are so small. So we have looked at some drugs that potentially don't cross the blood-brain barrier, and they have peripheral effects. Especially in our asthma model, we've looked at a couple of those that don't get into the brain, and those can fully rescue the asthma phenotype.
I guess I have one follow-up question, if I may. Because you say they have proven like also behavioral effects on mice, but are the different components, like other than the known ones like psilocybin, are they tested in human models already, or?
Yeah,
that's the head twitch behavior is one thing, but seeing what
it
actually does when you take it is probably another.
Yeah, that's a very good question. And one of the unique things about studying psychedelics is oftentimes psychedelic is in a person before it is in a mouse. And a lot of these, I think every one that we looked at in that panel was described by Sasha Shulgin in PCAL or TCAL. And the one of the drugs that we tested that was a fully efficacious anti-inflammatory was called 2CH. And that was, I think, one of the only ones that we tested that was anti-inflammatory that Sasha had reported there was no activity. But we think that's because it's just metabolized very quickly. But the vast majority of the drugs that we've looked at were all described in PCAL or TCAL.
Very interesting.
And
Thank
do
you.
have human effects.
All right. Thank you for your talk, by the way.
Oh, thank you.
Are there any further questions? I actually have 2 questions, but if someone wants to raise their hands at any moment, don't hesitate. I'm just wondering whether you can say something. So there is a little bit spoken about a theoretical heart risk of microdosing due to serotonin 2B agonism. What can you say to that regarding the models that you are testing?
Yeah, so it's something that we've looked at, but not really published yet. And I think that again goes to my hypothesis is that at least like a single large dose that's being used for the treatment of mental health is probably not an issue. It's really the sustained activation of the 5-HT2B receptors to a specific level over time that induces those valvulopic effects. And with something like microdosing, you're not, I don't think, this is just my personal opinion, I don't think that the 2B receptor is being activated to such an extent that it will induce those effects. We have submitted a grant to NIH to study that effect and sort of really delineate that, but it was not funded. The other type of thing that we've done is we've done chronic dosing of LSD in a rat model. And this was reported, I think, in our publication in 2012, where we were looking at chronic LSD on behaviors. But we took the hearts out of those animals and had them sectioned and had a pathologist look at them blinded to the treatment and could see no difference. These were animals that were treated for 3 months with LSD every other day, a 0.1 milligram per kilogram. And we didn't see anything abnormal in those hearts. So we haven't published those, but I'd say my opinion is that these drugs probably are not going to be sufficiently activating the 2B receptor at that level. That being said, there still could be a risk because nobody has really looked at it yet.
Right, but so based on the data you are talking about now, the protocol that you've been testing might be translatable to humans without risks for the heart, but needs to be looked into.
And we did one study where we were looking at renal inflammation, and we had implanted DOI in those same mini pumps. This was in a rat model, and we put a telemetry probe in those animals, and so with that, we're able to continuously monitor their blood pressure and their heart rate. And over the 2 months that we monitored the cardiovascular blood pressure and heart rate in those animals, we saw no abnormalities at the functional level, but we weren't specifically looking for a long QT or other effects. So just in my lab, we've done a few things to see if we could see something, and we have not seen a hit.
Okay, interesting. If no one has a question, I have a second question. Just it's a broader question, but interested about, how does this, in your view, translate to all the inflammation that's more linked to mental health disorders? Because it's interesting to see that, across the spectrum of psychedelics that are now being tested for depression, let's say, they range very differently on whatever you are testing regarding to anti-inflammation. So how would you connect those two?
Yeah, so that's something that as soon as we found these results, I'd always been wondering about. Being a neuroscientist myself, back before psychedelics became popular, I was trying to get people who were looking at neuroinflammation to see if they would be willing to test some low doses of DOI and do some scans, and I was just had to focus in the periphery. But there is about a 40% of patients with major depression have inflammatory biomarkers. And so there is a significant subset, and it could very well be that some of the therapeutic efficacy of psilocybin or other psychedelics would be to treat the inflammation that would be occurring in those brains. It's just a theory. We haven't looked at that yet. And unfortunately, we are looking in 2 different models of depression when we're doing our psilocybin studies, looking at the long-lasting efficacy. And in those studies, one of them is adolescent chronic restraint stress, where we do a 1 hour of restraint stress a day in female rats during adolescence. And when we stop that and they go into adulthood, essentially for the rest of their life, they exhibit these depressive-like behaviors. And when we give them a single treatment with psilocybin as a young adult, think we've tested now 7 or 8 weeks later, they don't have the depressive symptoms. They have a restoration of cognitive function. So we thought, well, maybe there's some inflammation in those animals that we could look at. But when we looked at the brains of those control animals for different inflammatory biomarkers, we didn't see any. So the model of depression that we're using didn't induce any neuroinflammation. I think maybe some of the more stringent models, like the chronic mild unpredictable stress, which is kind of like a PTSD model, it might. There was a collaborator that was studying the effects of DMT on PTSD-induced reactive oxygen in the brain, Parker Kelly, who is now a postdoc at UCSF. And he published, I think, 2 papers, I think two or 3 years ago, showing that DMT can reduce reactive oxygen species in the brain of rats that were exposed to PTSD-inducing conditions, predator odor response. So there's some indirect evidence from those experiments on the neuroinflammation.
Okay, and it's also really interesting because in your peripheral analysis, like DMT was ranking quite low, and then so it's a very diverse spectrum. Let's say you cannot talk about inflammation, and it's one thing. Like you have many different mechanisms, and every psychedelic might act differently on different mechanisms of inflammation and neuroinflammation.
Yeah, it could be. It's they all share some aspect of anti-inflammatory activity, but some psychedelics look like they're better for some types of inflammation than others, and so that's been interesting trying to sort that out.
Right. Okay. Are there any more questions? I don't see anyone raising their hands or anything in the chat. Would you maybe like to share something to finish, or are we gonna complete the presentation? I guess this is not an interview, so I don't need to ask you what do you have to say to young researchers or something. I mean, actually, there is one more question actually in the meantime. So go ahead, Alexander.
Sorry for the late question, but how far, or is there a timeline for when this would go into clinical trials in humans, or do you have an estimation about?
Good question. So my father and I and my collaborator at LSU formed a company a couple years ago to develop anti-inflammatories for the clinic, and those are going it's going fairly well. Our first indication is going to be an ocular inflammatory disease, and if things go well, we'll have our first clinical trial in patients at the end of next year. That we're now in the stage where we're getting a large amount of our lead candidate made, do the toxicology, do the GMP work. So this is a long process, but hopefully by the end of next year, we'll have something.
And why for ocular inflammation?
Part of it was I think, less concern from the FDA because it will be ocular, even though it's so low and there's no behavior. The FDA still would be concerned about systemic exposure and 5-HT2B. So hopefully we won't have to do those types of studies looking at the heart. And also the ocular indication, the clinical trial is only a few weeks. Whereas if we were to do something for asthma or for arthritis, we're looking at 2 years and very expensive. So our strategy is to get something in a short clinical trial, show it works, and then use that to generate the revenue to then go for some of these other treatments.
Okay, cool. Thank you.
But it's fun. It's been interesting getting into the biotech side of things as well.
Okay.
Yeah, so that's something else to look forward to. I think everyone is looking forward to the non-psychedelic psychedelics being tested in humans, and then psychedelics at subbehavioral doses being tested for inflammation.
Yeah, we're just now starting to test some of our non-psychedelic psychedelics in our depression models. So
okay,
just
that's gonna
starting that this week.
Okay. Interesting.
Is this the 2CH that you mentioned?
No. It's a third generation anti-inflammatory. So when doing the molecular modeling in the structure activity relationship, I think have identified some key residues that we could engage that would turn on the or maximize the anti-inflammatory effect. And it just so happened that when we do that, the behavioral effects went away. So we kind of, for one change, we hit two things, which we were very lucky. So we have these new drugs now, and we've tested them in several inflammatory models, but now we're going to look in this model of depression, see if it's effective there.
All right, and just one last question, sorry, because there's depression models that are looking at inflammation and depression, and I think we've known that PTSD is quite linked with neuroinflammation. So is it possible? I mean, I don't know about mice models. I think it's easier to, and I don't know if PTSD translates to mice models, and can we induce PTSD or what we model as PTSD in mice, and then and so then why study depression where you say that if I understood correctly, that it's not always, you don't always find inflammation in depression, but then PTSD seems to be a much more robust relationship between So my question
PTSD
is sorry.
The PTSD is a really robust in inducing inflammation, whereas I think in depression, just looking at peripheral biomarkers in the blood of, say, increased pro-inflammatory markers, there's 40 to 45% of patients with major depression have that inflammation. I don't know if there's any differential diagnosis or phenotypes associated with the inflammatory versus the non-inflammatory. And then it's also not known, did the inflammation cause the depression, or did the depression cause the inflammation? So there's still a lot we don't know about that. In the PTSD models in rodents, one that's used quite often is predator stress. So you'll take a mouse or a rat and you put it in a cage with a clear barrier, say with a cat, and stress it over time, or you get odor from a cat and you put it in a cage with that odor and it can't escape from it. And you do that a few times and it develops this almost PTSD type response. And those were the types of behaviors that were in the paper from Parker Kelly when he was looking at DMT and the reactive oxygen in the brain. He was doing that predator stress.
Right. And so is there a reason to test for the inflammation in depression models instead of PTSD models?
I think we've primarily been focusing on depression, just as it's more prevalent in the society. Depression that will affect so many more people.
Right.
And in terms of like the psychedelic assisted psychotherapy, the MDMA is primarily been used for that, which is a completely different mechanism in the brain. Although I think there are some clinical trials now using psilocybin for PTSD, and I don't think that the results of those have been released yet.
I know. Yeah.
So prior to the Hopkins and NYU studies coming out in 2016, we were really focused on looking at the effects of psychedelics on individual cell types in the brain. What were they doing to excitatory neurons? What were they doing to microglia? The gene expression response, sort of a more fundamental. And we're still doing some of that, but then once these depression studies came out, we started looking at, could we model the long-lasting effects? And we've been able to, in these models, show that a single treatment, and we've used psilocybin and a couple other drugs, a single treatment has long-lasting effects on functional plasticity and behaviors out to more than 100 days after a single treatment. And so we're now trying to look at what's the mechanism of that. What are the epigenetic factors underlying that? So that's where the research on the neuroscience side of my lab is sort of focused on now.
Right. Okay. Super interesting. Thank you.
Thank you. I think there are no more questions.
Okay.
That was really brilliant. Thank you so much. It was a great
Oh, you're
pleasure
welcome.
having you, Professor Nichols. I think I'm speaking for everyone here. Oh, there was one hand raising. No, no. Okay, people are applauding. So thank you so much, and we'll be looking forward to reading more of your publication and maybe having you over one more time in, after you've done this human studies and so forth to follow up on your research. Thanks a lot.
All right. Thank you. Thank you.
And,
Thank you.
Thank you to everyone, and see you then next time, in a month. I guess all the information will be on the website for the members.
And happy birthday, PSBE.
Happy birthday. Bye-bye.
Bye-bye.
Bye.